learners, welcome to this lesson where we want to look at comparison between plants and animals. This table shows the differences between plants and animals. First, most plants possess chlorophyll, which gives them their green color, so that the green pigment in plants is called chlorophyll, and it is very useful in the process of photosynthesis, that is the process of manufacturing food. On the other hand, animals lack chlorophyll pigment, hence they feed on already manufactured food materials. Hence, animals are called heterotrophs. The other differences between plants and animals is that plant cells have cellulose cell walls. Cellulose is a component that is found in the cell walls and it gives them a rigid shape. Animals lack these cellulose cell walls. They only have what we call a cell membrane enclosing the other cell contents. Plants respond slowly to changes in their environment, but animals respond quickly to changes in the environment. For example, when you touch a hot pan, you drop it immediately, so that is responding to the changes quickly. Plants are immobile, that is, they do not move from one part to another. They only move parts of their bodies like flowers, leaves, and even branches. But most animals move about in search for food, shelter, and mates. So animals move from one place to another. Plants lack specialized excretory organs because they have little or less metabolic activities in their cells. Metabolic activities are those activities or that processes that occur within their cells. So plants lack specialized excretory organs. Animals have complex excretory organs to excrete or to remove the waste products that come out of rapid metabolic processes within their cells. Let us go ahead and look at practical activities, that is collection of specimen. Since we have defined biology as the study of living things, therefore, for effective study, a biologist must have to collect some living things or some part of living things for observation and then analysis later. The living things or parts of living things that are used for biological study are called specimen. Some cases you may uproot a whole plant or some parts of the plant. So these are the specimens that will be used in the biological study. Biological studies always take place in laboratories. I believe you had one in your school. A laboratory is a building or a room that is designed and equipped with scientific apparatus used for scientific studies. Collection of living things, especially animals, may not be very easy, since some of these animals are not easy to catch, while some are quite dangerous. Knowledge on proper specimen collection and handling is very important. We will discuss some of the apparatus used in specimen collection. But before we discuss the apparatus, let us look at the precautions during collection and observation of specimen. Care needs to be taken so that we avoid injury during collection of specimen. So what are some of these precautions? One is that while collecting specimen for observation, a biologist needs to collect only the number of specimen you need. Do not collect more than you need. Do not collect excess. If you need 2, just collect 2. That will be enough, so that we do not destroy the organisms in our environment. Again, do not harm the specimen during capture or collection exercise. Do not injure them. Handle them with care so that they will be available for the next lesson. Do not destroy their natural habitat, that is the home for this specimen. So do not destroy their home. And after even collect them, you need to return them to their habitat if possible. Lastly, handle dangerous or injurious specimen with care. Such injurious specimen can be stinging plants or insects. So you will require the use of an apparatus called a forceps and even hand gloves in such cases. Finally, let us look at the apparatus used in specimen collection. The first one is a sweep net. It is used for catching flying insects. You know these flying insects, for example, butterflies, grasshoppers. So a sweep net is used to collect such specimen. And that is how it looks like. The other one is a fish net, from the term fish. So you should know that it is used for trapping small fish and other small water animals. It looks like this, but maybe you have seen a different one from this. Another apparatus is called a putter, and it is used for sucking small animals from rock surfaces or barks of trees. This is how a putter looks like. This is a tree, so this is a specimen that has been collected inside here. Pitfall trap is another apparatus, and it is used for catching crawling animals. You can see these animals that crawl on the floor like cockroaches. Another apparatus is called a pair of forceps, and this one is used for picking up small crawling animals like the stinging insect. Those insects that bite are called stinging insects. So a pair of forceps is used. Specimen bottle is the other apparatus, and these are the bottles used for keeping collected specimen, and they are of different sizes depending on the size of the specimen being studied. The other apparatus is called a magnifying lens. To magnify means to enlarge objects, and it is used to enlarge objects. A hand lens is a common magnifying lens used in the laboratory. The magnification power of the hand lens is always indicated on the hand lens. For example, times 10, times 5, times 8. This means the object has been magnified 10 times. Times 5, it has been magnified 5 times, and so on. The magnification power of a lens shows how many times the image will be enlarged compared to the object. That is a typical hand lens. Maybe you have seen one like this or different from this in your school laboratory. That marks the end of our lesson and complete this assignment. Goodbye.